Hello everyone. The topic for today's discussion is basic staining and mounting procedures for paraffin sections. Steps in staining and mounting paraffin sections are following. Number 1 is removal of wax with xylene, then hydration through alcohols. Third step is staining, then dehydration with alcohol. After that, clearing in xylene, and last, mounting under a cover slip. So you can remember it like rehydration should delay cause for morbidity. Rehydration, removal of wax with xylene. Hydration, hydration through alcohols. Staining, dehydration, cause clearing, then mounting under a cover slip. So rehydration should delay cause for morbidity. The first step is removal of wax. Wax is miscible in xylene alone. So first the sections mounted on staining rack, they are kept in an oven to heat at a temperature of 56 degrees Celsius for 1 hour so that wax is melted. Once wax is melted, the slide is dipped in Copeland jars containing xylene for 5 minutes. So after melting of the wax, the slides are kept in xylene. So removal of wax by xylene. It is kept for 5 minutes. The excessive heating in oven should be avoided. It may char the tissue. So after removal of wax, second step is hydration. As xylene is immiscible in water and will prevent staining. So take the solution section to a solution which will dissolve in both xylene and water, and that solution is absolute alcohol. So after xylene, the sections are transferred to absolute alcohol for 1 to 2 minutes. The solution will become opaque. If the wax has not been removed or some wax is not removed, then clear patches will be seen and such section should be returned to xylene for a further minute. So after xylene, the sections are put in the absolute alcohol for 1 to 2 minutes. The section is rinsed in second bath of absolute alcohol, drained and then taken to water. So 2 baths of absolute alcohol. If the water becomes cloudy, the second alcohol bath is contaminated with xylene and it should be changed. Now third step is staining. Here we will discuss HNE stain. The slides are then immersed in hematoxylin for 15 minutes in Copeland jar or rack. Then it is washed in running water. The arrangement of running water should be such that the water should be freely flowing and not showering directly on the slides. If water is directly falling on the slides, it may dislodge the section from the slide. So it should be washed in running, free flowing running water. Then third step is differentiation. The sections are then dipped in acid alcohol, that is 3% HCl in 70% ethanol, where they are agitated for few seconds and then washed in tap water. This will differentiate blue nuclei from rest of the field. We should examine under low power of microscope to ensure adequate differentiation and staining. So, differentiation with the help of acid alcohol. So on seeing in the microscope, if they are still undifferentiated, then return the section to acid alcohol. If over differentiated, then return to hematoxylin. Fourth step is bluing. When the sections are removed from acid alcohol, they are pink. Bluing is the process whereby the nuclei of the cells are accentuated to blue color in contrast to the pink background, and this is achieved by water containing few drops of liquid ammonia. So bluing is achieved by using water containing liquid ammonia, or it could be by using tap water, or lithium carbonate can be used. So bluing is important step. Bluing is the process whereby the nuclei of the cells are accentuated to blue color by using water containing liquid ammonia or tap water or lithium carbonate. After bluing, then counter stain is used. Transfer the slides to 1% aqueous eosin. In HNE stain, eosin is counter stain. For 2 minutes for counter staining. Then wash them in running water for 2 to 3 minutes to differentiate eosin. These are the steps of HNE stain. First, the slides are immersed in hematoxylin for 15 minutes. Then it is put under running tap water. Then differentiation with acid alcohol. Slides are put in acid alcohol and then washed in tap water. After that, bluing with the help of liquid ammonia, then counter staining, and again washing in running tap water. After staining, fourth important step is dehydration. Dehydration. Take slides through 3 changes of absolute alcohol. So dehydration by absolute alcohol, and we are using 3 changes of absolute alcohol, sequentially agitating for about 30 seconds in each. So we will use 3 jars of absolute alcohol, and in each we put in each jar for 30 seconds. Fifth step is clearing. After dehydration, clearing. Alcohol is then removed by dipping the slide in xylene bath. So again we are putting the slides into xylene. This takes about 5 seconds. Now what is clearing? Clearing means there should be no patches of water on the slide. How it is tested? It is tested by holding the slide against the light. Water will give opaque appearance if present. In that case, sections are to be dipped in alcohol once again before exposing to xylene, since alcohol is miscible in xylene and water equally well. Milky xylene suggests contamination, and xylene and alcohol should be replaced with fresh stocks. If xylene is milky, it should be replaced. Finally, the sections are transferred to second xylene bath. So, clearing by dipping the slides in 2 baths of xylene, second xylene bath before mounting. Now, sixth and final step is mounting. So, for mounting, we have to prepare, first we have to prepare a sufficient number of cover slips for sections to be mounted. They are kept ready. So, the preparation is, we should be ready with the cover slips. The cover slips used are rectangular. They should be free of dust. There should be no finger marks on the cover slips and should be placed on filter paper flat by holding the sides. Now the stain sections are removed from the xylene. After removing from the xylene, the excess xylene should be wiped off. Then we put 2 to 3 drops of mountain DPX. So what is DPX? DPX is diestyrene 80 dibutyl phthalate plus xylene. So diestyrene 80 dibutyl phthalate and xylene. 2 to 3 drops of DPX are put on the center of the slide containing section. The slide is then quickly inverted over the cover slip and brought down horizontally until the mountain is uniformly spread over the cover slip, under the cover slip over the entire section. Air bubbles, they can be removed by tapping a glass rod over the cover slip gently. And these air bubbles, they are formed if too little of mountain is applied. So the amount of mountain is important. 2 to 3 drops of mountain. It is put on the center of the slide containing the sections. The slide is then quickly inverted. So you have to invert the slide over the cover slip and then remove the bubbles. So this is the last step of staining, that is mounting. Thank you very much.